When you open your fridge, right, and you grab a carton of milk, you see the word pasteurized.
Yeah, everybody knows that word.
Right, and it's a word that it just implies this absolute safety, sterility, and pure objective science. You see that word on your milk carton, and you probably picture a meticulous, flawless scientist in a spotless white coat, just, handing down this pristine discovery to humanity.
Naturally.
But the man behind that word wasn't even a medical doctor.
Not at all.
He actually lost 3 of his own children to the very types of diseases he spent his whole life fighting. And as his private lab notebooks eventually revealed, notebooks he kept hidden for nearly a century, by the way, he engaged in massive public deceptions to secure his legacy.
It's a phenomenal contradiction. Today we're drawing entirely from excerpts of the comprehensive Wikipedia article on Louis Pasteur to reconstruct his life.
Yep, the whole picture.
And you quickly realize that the history of modern medicine, it just isn't a straight line of pure genius. It's messy, it is intensely competitive, and it's driven by deeply flawed, complex individuals who are just pushing themselves to the absolute limit.
Exactly. So our mission today for this deep dive is to look past the milk carton in your fridge and uncover the real Louis Pasteur. And to do that, we actually don't start in a medical lab at all, do we?
No, we really don't. We have to start with a young guy in the 1830s who was, honestly, a completely average student.
Like, surprisingly average.
Oh, yeah. Far from a child prodigy. Born in 1822 in Dole, France, young Louis was dyslexic and dysgraphic.
Right.
Barely scraped by in his early schooling. He much preferred, going fishing or meticulously drawing pastel portraits of his neighbors in the town of Arbois.
So he was kind of an art kid.
He was a highly visual person, totally obsessed with capturing exact physical details on canvas.
Which, that sounds like just a nice historical footnote, but that specific obsession with visual detail completely dictates his first major scientific breakthrough.
It absolutely does.
Because he eventually makes his way into the prestigious École Normale Supérieure in Paris. And by 1848, he's a chemist. And he gets pulled into this highly specific, incredibly frustrating mystery involving tartaric acid.
Yeah, and this is where the story really takes off. Because chemists at the time had 2 versions of this acid. One was derived naturally from the living ferments of wine.
Yeah.
The other was synthesized artificially in a laboratory. And chemically speaking, they were identical. They had the exact same elemental makeup.
Right.
But they behave differently when exposed to polarized light.
Okay, let's actually pause on the polarized light for a second for you listening, because that's not something most of us deal with every day.
Fair point, yeah.
Normally, light waves are, vibrating frantically in every possible direction. But if you pass that light through a special filter, you can block out everything except the light waves vibrating in one single flat plane. That is polarized light.
Exactly. And the chemists noticed that if you shoot this flat plane of polarized light through the naturally derived tartaric acid, the light twists.
Like it physically turns.
Yes, the plane actually rotates as it passes through the liquid. But if you shoot the same light through the synthetic lab-made acid, the light stays perfectly flat. It doesn't rotate at all.
Which makes no sense.
Right. The scientific community was completely baffled. If the chemicals are made of the exact same stuff, why on earth do they interact with light differently?
And this is where the dyslexic portrait artist steps in, right?
Yes, he brings that intense visual scrutiny to the problem. He takes the synthetic acid, crystallizes it, and actually puts it under a microscope.
And he notices something that every other prominent chemist had completely missed.
Yep. The synthetic crystals had these tiny microscopic asymmetrical faces on them. And more importantly, the pile of crystals was not uniform. Half of the crystals were angled slightly to the right, and the other half were angled slightly to the left.
Okay, let's unpack this. It's basically like looking at a massive microscopic pile of gloves.
Oh, that's a great way to look at it.
Like if you just glance at them, they all look like generic hand coverings with 5 fingers. But if you look really closely, you realize you have a distinct pile of left hands and a distinct pile of right hands.
Exactly.
They mirror each other, but you cannot perfectly superimpose a left glove onto a right hand.
That is the defining concept right there. So using a needle, Pasteur manually separated the left-handed crystals from the right-handed crystals under his microscope.
Wow, by hand.
Yeah. And when he dissolved the right-handed ones and passed light through, the light twisted to the right. The left-handed ones twisted the light to the left.
Oh, wow.
So the synthetic acid hadn't been twisting the light initially because it was a 50-50, what we call a racemic mixture today. The left and right twists were simply canceling each other out.
But the natural acid from the wine ferment only produced the right-handed version. It was purely one shape.
Which is huge. This proved, for the very first time, the concept of optical isomerism or molecular chirality.
Meaning shape matters.
Exactly. Pasteur demonstrated that molecules are not just a flat list of chemical ingredients. They have a physical, three-dimensional internal architecture. And honestly, this is the bedrock of modern medicinal chemistry.
Right.
Today, we know that human cellular receptors act like locks and drug molecules act like keys. If a drug molecule has the wrong 3D shape, like if it's a left-handed key for a right-handed lock, it won't work. Or worse, it could be toxic.
That is incredible. But a chemist staring at microscopic crystal asymmetry feels very far removed from vaccines and diseases.
That does seem like quite a leap.
But that visual understanding of the natural world naturally pushes him toward biology. Because if you are the guy who solved the wine acid mystery, you kind of become the go-to person when local industries have problems with their liquids.
Right. Word gets around. And that happens in 1856. He's working in Lille, which is a major industrial hub, and a local manufacturer approaches him with a pretty devastating problem. His massive vats of beetroot alcohol are going sour.
Just totally spoiling.
Yeah, and it's ruining his business.
And the scientific consensus at the time, led by heavyweights like Justus von Liebig, was that fermentation was purely a chemical breakdown, right? Like dead matter just decomposing, like iron rusting.
Exactly. But Pasteur brings his microscope. He doesn't just look at the chemical composition of the souring beetroot juice. He looks at the physical shapes suspended inside it.
There's the artist eye again.
Yes. He observes the yeast cells. And he realizes these cells are budding, they are reproducing. The yeast isn't just a chemical catalyst, it is a living organism.
Wow.
Fermentation is actually a biological function of a living creature consuming sugar and producing alcohol.
Which means if the vat is going sour, it's not a chemical imbalance, it's an invasion.
Precisely. He finds a different, much smaller microorganism swimming in the spoiled vat's lactic acid bacteria. They had basically invaded the vats and were out competing the yeast, producing sour lactic acid instead of alcohol.
So the immediate industrial problem becomes, how do we get rid of the invaders without destroying the product? I mean, you can't just dump bleach into wine or milk.
No, nobody wants to drink that.
Right. So in 1865, he patents a method. He figures out that if you gently heat the liquid to between 60 and 100 degrees Celsius, it's hot enough to kill the contaminating bacteria, but not so hot that it ruins the flavor of the beverage.
And pasteurization is born.
Saving the French agricultural economy.
He really did. But, you know, this industrial fix triggers a massive philosophical earthquake.
Oh, yeah.
Because if microscopic living organisms are responsible for spoiling wine and milk, where are they coming from?
Right. And you listening are probably very familiar with the spontaneous generation debate. It's easy for us today to look back and scoff at the idea that bugs just magically materialized out of dead matter.
Oh, it sounds ridiculous to us now.
But at the time, this was entrenched, serious science.
Wait, let me just clarify. People in the 1860s seriously still believed bugs just magically poofed into existence from thin air.
Oh, absolutely. It was fiercely defended scientific doctrine, championed by highly respected figures like Félix Archimede Pouchet.
That's wild.
Pouchet and others believed that the air itself possessed a vital force.
A vital force. Okay.
So if you left a bowl of beef broth on a table, and it went cloudy with bacteria a few days later, they argued the life was generated spontaneously from the nutrients and that vital force in the air.
Magically.
Right. But Pasteur argued the exact opposite. He said life only comes from existing life. The dust floating in the air is actually carrying the microscopic parents of those bacteria.
So the French Academy of Sciences essentially sets up a scientific duel. They offer the Humbert Prize in 1862 to whoever can definitively prove their case through rigorous experimentation.
And Pasteur responds with one of the most elegant experiments in scientific history, the Swan Neck flask.
Yes, classic.
He takes a glass flask, fills it with nutrient broth, and boils it to absolutely kill anything currently living inside. But instead of just corking it, he heats the glass neck of the flask and pulls it out into a long, thin S-shaped curve.
The geometry here is the genius part. The end of the tube is completely open to the room. So Pouchet's vital force, the oxygen, the air itself, can freely flow down the tube and into the broth.
Exactly, the air gets in.
But dust particles are heavy. As they drift into the open tube, gravity catches them in the lowest dip of that S-curve. They literally can't float up the second half of the curve to reach the liquid.
And the result was undeniable. The broth inside the Swan Neck flasks remained crystal clear and sterile, not just for days, but for months and years.
Wow.
No spontaneous generation occurred, even with full access to the air. But the absolute mic drop moment was when Pasteur simply tipped the flask.
Oh, I love this part.
He let the sterile broth run down into the curve, touch the accumulated dust, and flow back into the main chamber. Within days, the broth was swarming with life.
He conclusively proved that the invisible threats are environmental. They are riding on dust. They have parents. And this establishes the foundation for what's about to be a very dark, very intense chapter in his life. Because if invisible, living invaders floating in the environment can infect and kill a vat of wine, it is a terrifying but very logical next step to wonder if the exact same process is happening in the blood of animals.
Right. That theoretical leap from spoiled beverages to the germ theory of disease was forged in an era of unimaginable personal grief.
Yeah.
Between 1859 and 1866, Louis Pasteur and his wife Marie lost 3 of their 5 children.
3 of their 5.
Jean died at age 9, Camille at age 2, and Cécile at 12, and all 3 succumbed to typhoid fever.
I mean, you just cannot overstate the psychological weight of that. To be the man who is literally mapping out the existence of invisible living killers and to be entirely powerless to stop those same killers from taking your own children.
It's heartbreaking, and it completely shifts his work from, industrial troubleshooting to an absolute crusade against disease.
Right. But his first major battlefield in that crusade wasn't human medicine, was it?
No, it was another agricultural crisis. In 1865, the French government desperately asked him to investigate a plague wiping out the silk industry in the south of France.
Silkworms.
Millions of silkworms were dying from 2 mysterious conditions called pébrine and flasherie.
Now, keep in mind, Pasteur is a chemist who studies crystals and yeast. He knows absolutely nothing about silkworms.
0.
But he applies his methodology anyway. He sets up a lab and spends 5 grueling years studying these insects.
And it is crucial to note that he floundered at first. He wasn't instantly successful. He actually initially rejected the idea that the silkworm disease was parasitic, even when his peers suggested it.
Really?
Yeah, he spent a long time chasing the false assumption that the 2 separate diseases were just like variations of one ailment.
But his persistence pays off eventually. He realizes that the disease is characterized by these microscopic corpuscles inside the worms, and more importantly, he proves it is hereditary. An infected mother moth passes the parasite directly into her eggs.
So he devises a pretty ruthless but scalable solution. He instructs the farmers to let the female moths lay their eggs. Then they crush the mother moth into a pulp and examine her under a microscope.
Wow. Okay.
If they spot the corpuscles, they immediately incinerate all of her eggs. If she is clean, they keep the eggs for the next generation.
It's basically the invention of a microscopic quality assurance assembly line.
That's a great analogy.
And it worked. He saved the French silk industry, but far more importantly, he proved that you could control an epidemic in an animal population by isolating and destroying the specific microorganism responsible.
Which is the massive bridge to immunology. He has proven that germs cause disease. So now the focus shifts to how do we train a body to fight them off?
Right. Saving silkworms is one thing, but how does he make the jump to actively saving livestock and humans with vaccines?
Well, by the late 1870s, Pasteur is studying chicken cholera. And the breakthrough here actually stems from a fortuitous oversight.
A mistake.
Pretty much. He was preparing to leave for his daughter's wedding, and his assistant, Émile Roux, just left some cultures of the deadly cholera bacteria sitting out on the lab bench, exposed to the air.
And when they eventually come back and inject chickens with this old, neglected culture, the chickens get a little sluggish, but they don't die. They fully recover.
But the real revelation comes when they later inject those exact same chickens with a fresh, highly virulent, lethal batch of the bacteria.
And nothing happens.
The chickens are completely unaffected. They had developed immunity.
Unbelievable.
Pasteur realizes that leaving the bacteria exposed to oxygen over an extended period had slowly degraded its metabolic pathways. It starved the bacteria just enough to weaken it, and he coined the term attenuation for this process.
The mechanism there is fascinating. Because the bacteria is still alive, so its physical 3D shape is intact, meaning the chicken's immune system can still learn to recognize it and fight it, but it's too weak to actually overrun the host. It's essentially a low-stakes sparring partner for the immune system.
That's exactly what it is. And having proven this attenuation method on chickens, Pasteur aggressively sets his sights on a much bigger target, which is anthrax.
Oh, yeah.
This was a brutal bacterial disease, totally devastating cattle and sheep across Europe. And this is where the historical reality of 19th century science gets intensely political. Pasteur was locked in a bitter rivalry with the brilliant German physician Robert Koch.
Right. Koch was the one who had actually isolated the anthrax bacteria first.
Yeah.
He was incredibly methodical, and he was highly critical of Pasteur's flashy, less rigorous methods.
Oh, they couldn't stand each other.
And remember, this is shortly after the Franco-Prussian War. There's intense national pride at stake.
Yeah.
France versus Germany.
Huge pressure. Pasteur felt immense pressure to deliver a French victory. He publicly claimed he had developed the successful anthrax vaccine using his signature oxygen attenuation method.
Okay.
And he agrees to a massive, highly publicized field trial in 1881 at a farm in Puy-le-Fort. It was a total media circus.
I can imagine.
He took 58 animals, he vaccinated half of them, left the other half unprotected, and then injected all of them with lethal anthrax. He boldly predicted the unvaccinated would drop dead and his vaccinated animals would survive.
And he was right. It was a total triumph. The vaccinated animals lived. The world hailed him as a scientific savior.
Yeah, he was a hero.
But this is where the marble statue shatters a bit, because Pasteur had left very strict instructions with his family to never, ever let anyone read his private laboratory notebooks.
Right. And they kept that secret until 1971. When historian Gerald Gayson finally analyzed those notebooks in 1995, the medical history world was absolutely stunned.
I bet.
The notebooks revealed that at the Puy-le-Fort trial, Pasteur absolutely did not use his oxygen-attenuated vaccine.
Because his oxygen method wasn't working reliably for anthrax. So behind the scenes, his assistants, Roux and Chamberlain, had quietly turned to chemical inactivation. They treated the anthrax bacteria with potassium dichromate.
Exactly. Potassium dichromate is a harsh oxidizer. Instead of just starving the bacteria with oxygen over time, this chemical forcefully alters the surface proteins of the bacteria, crippling its ability to replicate while preserving enough of its structure to trigger an immune response.
But the scandal isn't just that he used a chemical. The scandal is that a rival veterinarian, a man named Henri Toussaint, had been actively working on chemical inactivation to create an anthrax vaccine.
Yep.
Pasteur publicly dismissed Toussaint's work. Then, when his own method failed right before the biggest trial of his life, Pasteur secretly used a chemical method remarkably similar to his rivals, passed it off as his own original oxygen method, and just absorbed all the global glory.
He really did.
Here's where it gets really interesting, though.
Yeah.
Isn't that just straight-up intellectual theft? Like, he used a competitor's recipe to win a public bet and took all the glory.
Well, it is a stark look at the human ego, for sure. By modern standards, claiming another lab's methodology as your own to win a public trial is a severe ethical breach. But we must contextualize it within the pressure cooker of that Koch-Pasteur rivalry. Pasteur was a showman, yes, but he was also a pragmatist. He genuinely believed that securing funding and public trust for the germ theory was far more important than transparent methodology.
So the ends justify the means.
In his mind, yes. He secured his legacy through deliberate deception because he believed the ultimate outcome saving lives justified it entirely.
And if we are talking about agonizing ethical boundaries, his final major triumph takes us right to the absolute edge. Rabies.
Oh, this is so fun.
In the 1880s, a rabies diagnosis was a guaranteed horrific death sentence.
And scientifically, it was a totally different beast than anthrax. Anthrax is a large bacteria. You can see it under a microscope. Rabies is a virus.
Right.
It was entirely invisible to 19th century technology. They didn't know what it looked like, but Pasteur and Roux deduced that whatever the pathogen was, it concentrated in the central nervous system.
So to build a vaccine, they have to cultivate an invisible enemy. They begin injecting the virus directly into the brains of live rabbits. When the rabbit dies of rabies, they extract its spinal cord.
And here we see the evolution of their attenuation technique. They took these highly infectious rabbit spinal cords and hung them inside sterile glass flasks containing chemicals to absorb the moisture.
Oh, okay.
As the spinal cord slowly dried out over several days, the viral envelope began to degrade. A spinal cord dried for 14 days was very weak. A freshly extracted cord was lethal.
So they formulate a schedule of injections, starting with the weakest 14-day dried tissue and slowly building up to the most virulent. They test it on dogs first, but testing it on humans is a completely different ethical universe.
It really is. And that moment arrives in July 1885. A 9-year-old boy named Joseph Meister is brought to Pasteur's laboratory.
Just a kid.
He had been viciously mauled by a rabid dog. The boy is bleeding, and without intervention, he is going to die a terrible, painful death.
I mean, you listening, put yourself in Pasteur's shoes for a second. You aren't a doctor. The animal trials are incredibly sparse, and you have a dying child sitting in front of you. Do you risk jail and the child's life on a hunch?
It is an excruciating calculation. Pasteur consulted with licensed physicians who agreed the boy's case was hopeless. To protect himself legally, Pasteur had a licensed doctor, Jacques-Joseph Grancher, actually hold the syringe and administer the injections.
So Grancher takes the legal heat.
Right. And over 11 days, they gave young Joseph 13 inoculations, each one containing stronger and stronger strains of the virus.
And Joseph Meister survived.
He did.
He never developed rabies. It was an unprecedented medical miracle. People began flocking from all over Europe to get this treatment.
But once again, the secret notebooks cast a long shadow over the triumph. When Pasteur published his results, he claimed he had perfected the vaccine by testing it on 50 dogs before ever attempting it on Joseph Meister, ensuring its safety.
Let me guess, the notebooks say otherwise.
The notebooks tell a very different story. He had only tested that specific protocol on 11 dogs.
Wow.
And much more shockingly, Joseph Meister was not his first human subject.
Wait, really?
Pasteur had secretly overseen the administration of early versions of the vaccine on 2 other humans prior to Meister. One patient survived, but likely didn't actually have rabies to begin with. The other patient died of rabies.
Wow. So he just buried those failures.
He completely buried them. He presented a sanitized, highly confident version of his research to the public. The tension here is immense.
Yeah.
His methods were reckless, highly secretive, and today they would be profoundly illegal.
Oh, absolutely.
Yet that specific agonizing gamble on a 9-year-old boy worked. The success of the Meister case led directly to massive international fundraising, which built the Pasteur Institute in 1887.
Which changed everything.
It did. That institute became the global epicenter for infectious disease research, establishing the protocols for modern vaccine manufacturing that have literally saved countless millions of lives.
It is an incredible journey. We started with a young boy drawing pastel portraits in Arbois. We followed his eyes as he manually separated microscopic left and right-handed crystals. We watched him redefine fermentation in beetroot vats, map out the geometry of a Swan Neck flask to defeat spontaneous generation, and hunt down parasites in silkworms. He challenged the greatest medical threats of his day, armed with ruthless ambition, secret methods, and an unyielding belief in his own vision.
Yeah, that sums him up.
The next time you grab a carton out of the fridge and read the word pasteurized, remember that you aren't just looking at a sterile heating process. You are looking at the legacy of a deeply complex, fiercely determined human being who dragged medicine out of the Dark Ages.
And to leave you with one final, truly awe-inspiring piece of context to mull over, something that reframes everything we just discussed.
Oh, lay it on me.
In 1868, when Louis Pasteur was just 45 years old, he suffered a severe stroke.
Really? At 45?
Yes, and it permanently paralyzed the entire left side of his body. Everything we covered in the latter half of this deep dive, the agonizing 5-year study of silkworms, the high-stakes public anthrax trials at Puy-le-Fort, the meticulous extraction of rabbit spinal cords for the rabies vaccine, the founding of his global institute, he accomplished all of that while severely physically impaired.
Are you kidding me?
For the most critical decades of his career, he was essentially working and changing the world with only one functioning hand.
It is almost impossible to comprehend. Just the sheer, unstoppable force of human will required to navigate that murky historical landscape, even when your own body is fighting against you. Thanks for joining us on this deep dive. We'll see you next time.